happen the frozen filling. Okay. So all those things can affect to the quality, the safety, and shelf life, as we mentioned about the process of product here. And also finally, when they have the perfect match in terms of these three, of course we can generate higher income. Okay, with less waste and more profits. Okay, so that's why kalau sekarang ni kita ada fokus on SDG, Sustainable Development Goal. Okay, so yang mana kita fokus on the make it a profit and sustain. Okay, in terms of the in terms of the sustain, in terms of the controlling or maintaining the quality of the environment and also the product itself. Okay. So how can you determine the fish freshness? Okay, ini saya rasa you guys sudah faham. Okay, macam mana untuk kita menilai. Okay, kalau you go to the fish market, also you can identify which one is the good, which one is the spoiled fish. Okay, when we talk about the fresh fish, of course, it has the clear eyes. Okay, this is black, has a black pupil, and also the fur. And when we touch the texture, also it bounce. Okay, kalau isi ikan itu you touch and it bounce. Kalau this they maintain to the degree of the physical touch, and then it is not fresh. Okay. And also, of course, the gills are bright red. This is the common characters with the clear slime. Okay. Slime ni disebabkan apa? Sometimes slime apa kita cakap dia punya mucus ya, ada lendiran yang tidak ikan tu. Okay. Sometimes because of the spoilage, or sometimes because of the stress. Bila ikan itu stress, okay, when during the handling, the fish can easily get stressed. When the stress happen, it can release the slime, okay, in the overall in the body, ataupun release at the body lah for that. Okay, and the spoiled fish, of course, it has a soft, discolored flesh, strong odor, and also you can have like the rancidity smells. Later on I will explain. Okay. Okay, this is the characteristics of the fish freshness. Okay, this the fresh and the spoiled fish characters. The eyes, I think this is easily general characteristic that you can understand. The gills, the flesh, and the belly itself. The belly impact means that bila you guys keluarkan the perut kan, okay. So it still have the good structure of the belly and the intestine structure. Okay, means that when it has a good structure, it's still in the fresh conditions. Compared to the sometimes they have open rupture. The viscera is protruding and so on. Okay, and the muscle tissue, the vent, okay, and also the odor and also the color. The color is easily to be identified. Okay. All right. So we have go for the introduction part. Okay. So let me elaborate on what causes fish to spoil. Okay, there are three basically. About the first thing is about the bacteria. Okay, when we talk about the bacteria, of course, we have the growth of the bacteria will cause the spoilage. Okay, where the microorganisms on the skin, gills, intestines rapidly invade flesh after the death, and this can cause the off flavor, and finally the bacteria also can release the toxic compounds. Okay, the bacteria will invade to the cells, invade to the tissue, muscle tissue, and it can cause the spoil to the fish. Okay, the second one is because of the enzyme. This is the we call it usually the autolysis lah. The autolysis is a natural and digestive enzyme in the fish muscle continue to function after death. Okay, meaning kita dalam badan kita, all the living organisms we have our enzymes. Enzymes is produced. Okay, misalnya kalau kita makan protein, the enzyme, the protease will help in digesting the protein. Okay, kalau kita makan the carbs, maybe the amylase. Okay, and so on. So similar with the fish also have the enzymes. Okay, and even after that also the enzyme is still functioning. Okay, for the certain storage for the certain period. Okay, and it helps in breaking down proteins and also softening. Okay, all right. So the third one, the cause the fish spoil is because of the handling. This is the most critical, okay? Because sometimes we think we are trying in the aquaculture, okay? Kita sudah pakannya sudah perfect, the optimum. We try to get the best fish. We try to culture in a very good condition. We try to optimize all the water quality, maintain the water quality, give the best. Okay, but suddenly, when at the end, when the final stage, when we are doing the post harvest, okay, the handling is rough, definitely it will affects to the spoilage. Okay, it affects to the low quality of the fish. Okay, so it's such a waste lah. We have spent many months. Okay, kalau tilapia culture-nya like about 4 to 6 months. So at the end, the quality drops because of just one thing at the last stage on the handling. Okay, so you have to be careful in terms of the from the beginning, from the culture up to the end consumers. Okay, how you handle the fish. Okay, what type of the net you're using. Some of the net you might kill or you might affect to the quality of the fish. Okay, and some of the net like the trawl net and so on. Okay, sometimes kalau di sini kita bilang ni pukat tunggal, pukat tarik yang kalau pukat tunggal itu kalau itu netnya yang yang banyak yang sekali itu. Bila pukat itu sometimes they affects on the gills. Okay, so bila dah compact and then that affects on the quality of the fish. Okay, so when the heat and rough handling it cause the high temperatures, and of course when it comes to the high temperature accelerate, the bacteria will grow. Okay, and enzymes will functions. Okay, continue to actively functions, and it causes the physical damage. Okay, of the destruction of the tissue, muscles, and so on. Okay, so these are the mechanisms like a bit detailed. Okay, in terms of the autolysis, the enzymes, because of the presence of the protein, the lipase is more on the lipids. Lipid liquidations, okay, to degrade the lipid or the fats, okay, and bacterial source, okay, this is on the contaminations, okay, bacteria will grow at the ambient temperature, microorganisms as well, the fungus, okay, yeast, mold, and other microbes, is growth on the surface or inside the fish, and the critical also is part, okay, the oxidation. What is oxidation? Basically, oxidation is the decomposition like that. The protein is oxidized when react with the oxygens. Okay, when those react with the oxygens in the air, it causes the oxidation. Okay, for example, lipid when react with the oxygens in the air, it can cause the lipid oxidation. When lipid oxidation happen, and afterwards it can cause the spoilage of the fish. Okay, and this oxidation, for example, if the lipid, okay, the fat, okay, when react, it can cause the rancidity. The rancidity might be happen, and also the discoloration and also flavor. Okay, so sometimes how we want to prevent or how we want to solve these problems. Okay, for example, the oxidations when you froze the fish. Okay, you boleh nampak different colors. For example, bila you letak frozen you freeze the fish in the freezer, the color might be pale. Okay, the pale happen because of the less oxygen. Okay, in the freezer, and also it can cause the low oxygen. Okay, there, and if you throwing kalau letak di luar semula, okay, the color might be different. Why? Because afterwards, when it reacts with the oxygen in the air, then it has in terms of improving the color. Itu usually we call as a blooming. Okay, the blooming. Blooming is the reaction of the oxygen, okay, in the meat products. Okay, and then it enhances the color back lah because of that thing, myoglobin lah. Okay, that is how the mechanism happen. Okay, and the rancidity, because of the fats and protein rancids, okay, because of the hydrolysis happen, and also because of the enzymes and the microbes, okay, react. Okay, and the Rhigomatitis is happen after that. Okay, saya rasa you guys are familiar with the term for the rhigomatitis. Rhigomatitis is where the muscle stiffening. Okay. Bila muscle stiffening, sometimes you can see the contractions of the muscles. Okay, bila contractions of the muscles happen, this rhigomatitis basically happens for at a certain period. Okay, for example, kalau ikan just a normal fish, ikan kalau ikan mackerels and so on, it happens like within 8 hours, dalam tempoh 8 jam. Okay, 8 to 12 hours lah usually. It depends on the temperature, the storage temperature and so on lah. Okay. Jadi kalau macam bila rhigomatitis berlaku, what happen? Because when stiffening of the muscles, the texture akan jadi keras. Okay, it will become hard in the texture. Okay, so bila hard, okay, the muscle stiffness will become hard, and then it can cause the sensory is not in the good quality. Okay, alright. So the proper handling can help in terms of prolong the pre-rhigo and the post-rhigo. Okay, sebelum berlaku rhigomatitis, dia ada pre-rhigo here and also the post-rhigo. Okay, what kalau rhigomatitis is not encouraged to be consumed at this stage? Okay, usually the consume before this or afterwards lah. Okay, usually kalau ikan, okay, just for information, kalau ikan usually kalau sashimi, okay, sushi and so on, we consume within before pre-rhigo, after slaughter the fish, and then we can consume. So that's why kalau the fish usually kalau sashimi, we have to consume less than 2 hours. Okay, bila dia cut, we need to consume. Why? The first thing is after 2 hours expected to be a rhigomatitis, it can cause the tough texture. Okay. The second thing adalah after 2 hours, this is the critical, the contamination of the bacteria. Okay, so kalau sometimes kalau you buat makanan, cakap consume less than 2 hours, consume less than 1 hour. This is why, because it affects on the bacterial contamination and the quality of the products. Okay, alright. Sama juga kalau you guys makan daging, okay, sapi. If you consume the sapi, so mungkin ya musim Idul Adha ya kan, musim Idul Adha, you after slaughter korban, after slaughter cow, and then you can directly cook the meat, and then this stage you can cause the tender. Okay, dagingnya lebih enak, lebih bagus and manis. Okay, the sweet. Okay, at this stage. Or else after the rhigomatitis. Usually kalau rhigomatitis untuk fish is about 8 to 12 hours. Okay, it depends on the species of the fish, but sometimes it can be for salmon it can be 16 hours. Okay, tapi kalau for the beef product, for the sapi, it can be 1 to 2 days rhigomatitis. So actually it depends on the type of the animal. Okay. So, why we have to consider that? Because everything is related to the shelf life. Okay, because the shelf life is after because if the product is spoiled, okay, it's no longer acceptable for the consumption. Okay, so that's why we have to consider how the commercial planning, okay, remaining the shelf life. Okay, either you are going to make into the processed food, either you are going to make how we arrange the distributions of the product, okay, and manage the stock rotation. Okay, this is how the critical managing or the preparations in this planning. Okay, And then the fresh versus frozen. Of course, kalau you ask the customer, you will prefer the fresh product because the fresh is more sweet. The fresh is more, they have a tender flesh compared to the frozen. Okay, but sometimes kita have an abundance of too many fish. Okay, so kalau with too many fish, sometimes we cannot sell. Okay, on the same day. So there's no way. So that's why we make it into the frozen. Okay, the function of the frozen actually basically is to extend the shelf life. When it is frozen, the bacteria will become less active or inactive. Okay, and also the enzymes also will slow at this stage, the enzyme activity. Okay, and of course the QC, the quality control. Okay, the quality control is the established used by the day to reduce the waste and maintain the consumers. So kalau you pergi dekat industri, you tengok they every batch, setiap batch pasar phase 2 they have the QC. Okay, they will randomly collect the samples, okay, and analyze for the quality control. Okay. So what are the factors, effects or influencing the shelf life? The raw material and the quality, raw material quality and age. Of course, the age. Okay, we can because when we talk about the age, it because of the metabolite. Okay, in our body we have the metabolisms. Okay, the metabolisms it depends on the age as well. Okay, it depends. Kalau fish, it depends on the stage. Okay, they have the juvenile, they have fry, they also have the adult fish. Okay, usually at the young stage, okay, the juvenile they have a high metabolism. Okay, itu yang sama dengan humans. Okay, kalau you guys anak-anak muda, okay, students, the metabolisms are active compared to the adult one, or compared to the old people. Because when it comes elder, okay, the metabolism is decreased. Okay, and then the seasonal variation and raw material history. Okay, where is the place? Where is the raw materials? Okay, yeah, you can get the supply. Okay, this is a factor how quickly a product will deteriorate. Okay, and next one is the process of treatments and handling. So type of the processing, either it is usually fresh, frozen, or into the processed products. Okay, or you can make, sometimes you can make a smoke. Okay, kalau di sana saya lihat banyak yang ada ikan bandeng ya. Kalau ikan bandeng they make it into the smoked fish. Okay, and also sometimes they make into the make it proper packaging. Okay, during the handling significantly influence the shelf life. And the final one is the bacterial contamination and storage conditions. Post-processing contaminations and inadequate storage temperature. Okay, this is can cause the shelf life failure. How you the shelf life, how you process, and then sometimes when they have a rough handling, it can cause the contaminations of the bacteria. Okay. So when we talk about the spoilage, a fresh is original characters. This is the keyword. Okay, the original characters remain unchanged. Okay, kalau spoil is of course they have exceed the limit ataupun limit acceptability to unacceptability. Okay, and this can cause the signs like transformations, discolorations, off flavor, odor, and also the gas productions. So when we talk about the physiochemical changes, based on the color change, the failure or discoloration, the odor developments, the smells, sometimes it causes sour, putrid, or ammonia-like of odors. The eye and gill changes, this is the comments, and also the texture softening, this is based on the texture when you are try to check on the bounciness of the products. Okay. So what happens after that? The first one is nucleotide breakdown. Okay. So students, which of I think maybe you have learned about the nucleotides. So which of this one, the fifth, of the components that can have the good flavor or the umami taste. Either the ADP, AFP, IMP, hypoxanthine and also inosine. Which one do you think? Ada idea?
IMP, IMP.
Ada yang mau jawab?
Dipta.
Silakan.
I think IMP. IMP.
IMP, betul. So when we talk about umami, so when we talk about the umami taste, of course we have the components that rich is at the umami, the IMP, inosine monophosphate. Okay. So, but we have to know the component, the breakdown of this molecular is happens rapidly. Okay. After ADP breakdown, the phosphate released to the ADP, AMP, the IMP. Okay. But afterwards, after IMP, this is the umami. But afterwards, when we talk about the hypoxanthine and so inosine, this is the flavor precursor. Means that this can bring to the bad or the unpleasant flavor. Basically, hypoxanthine and also inosine. Okay, later on, I think there's a slide for that explanation. And the second one is the rigamentis happens, as I mentioned, the thickening of the muscles within a few hours, depending on the how you catch or depending on the handling process. Okay. So this is the flow of the rigamentis. We have the pre-rigor. Whereby when we talk about the pre-rigor, is the fish is still flexible, and this is the good and the best quality that you can consume. Okay, that's why people like sashimi, sushi. At this stage, the taste or the flavor, the texture is very soft, tender, and also it has a sweet taste, basically, yeah. And then after that, going to the next one is the rigor answer, the full rigor. When we come to this stage, the full rigor, it can cause the tough texture, okay. And at this stage, it's not encouraged or we don't consume the fish, okay. So varies from 1 hour, actually a lot of mackerel, sardine, about 8 to 12 hours lah. Okay, sometimes it can be 24 hours. It depends on the fish. The salmon can be 16 to 24 hours. Okay, actually it depends also on the temperature of the storage and also the handling process. So when we talk about the handling, the bad handling it can trigger or initiate the full rigor. Okay, faster compared to the good handling process lah. Okay, and after full rigor, the resolution, the post rigor. Okay, this is muscle soften again. At this stage, enzymatic degradations. Okay, when enzymatic degradations, of course, at this stage also compare the post rigor and the pre rigor. Of course, the pre rigor has a more sweetness compared to the post rigor. Okay. So the effects of the rigamentis, it can cause the toughness, high grip loss, okay, during the experience, toughness for the towing, produce a large amount of grip, okay. Gapping, kalau gapping kita lihat di sini ya, dia ada di sini ada gap ni. Sebab dia punya putih dia makin besar, the gapping wide. So gapping is where the gapping fluid separate and slits, they appear on the surface. So this is happen because of the muscle group yang tadi will happen about the rigamentis, here muscle contract and relax. So bila muscle contract and relax, they have consequence to have the gapping. And the shrinkage of the proponent delays, okay, the muscle contraction during the rigor, reducing the yield and the appearance. Okay, so kita betul ada difference. This is the glossy, the smooth or the fillet during the pre-rigor, and this is the how it happened on the onset of the rigor. Okay. Okay, basically, when we talk about the fish polish, there are 4 phases. Okay. This is the 4 phases. The first phase one, the phase 1 and phase 2 is because of the autolytic changes. This is happened because of the enzyme activity. Okay, the phase 1 is the initial autolysis when after that, okay, they will, okay, when the fish is dead, okay, and then they will go to the 4 stages of the fish spoilage. The phase 1, okay, the initial autolysis, whereby it started the enzymes to initiate, okay, the autolysis, okay, at this stage, okay, and continue with the second stage, which is the autolysis, okay, whereby this autolysis have the loss of the natural flavors and it started to have the odor, okay, but this stage is still can be consumed, okay, because just initiate, okay, and continue for the autolysis at this stage, okay. Okay, but when we talk about this, the third and fourth stage, the third is started by the bacteria. Okay, bacteriological changes happen. Okay, so at this stage also is not encouraged to be consumed, but still can be. It's not encouraged, okay, but still can be consumed. Okay, okay. This is the sign of the spoilation. We can see the appearance, smell, the acidity, the unpleasant smell, okay, and the texture becomes tough and so on. And the last one is the final stage, the fourth one is spoiled. And this is the serious problem when they have a fourth stage, okay, so it is not encouraged to be consumed. Why? Because it's inedible and harmful. Why it is harmful? Because it produces the histamine, the high histamine. Okay, the histamine, where it comes from the histamine because of the nucleotide degradations, the breakdowns of the nucleotide degradations. Okay, and act with the bacteria, it can produce the histamine and also the biogenic amine. Okay, so this is the stage that is not encouraged to be consumed. Okay, so how long it takes? It depends on the species. It depends on the size of the fish. It depends on the age. It depends on also on the conditions, the temperature, the presence of the environment itself. Okay. So sometimes the spoilage, okay, for example, it can this spoilage happen like 2 to 3 days. Okay, so it has a rapid spoilage. Okay. Sometimes for the salmon, for example, it can be like spoilage after a week. Okay, it depends where do you store the fish. Okay. So when we talk about the spoilage mechanism, I think this is on this is the general, okay, of the idea because of the autolytic spoilage. Just now I mentioned the phase 1 and phase 2 becomes the softening of the flesh, and the microbial growth spoilage. This is the phase 3 and 4. Okay, because of the bacteriological changes happen, it can cause the degradation in most fish life. And the final part is the oxidation. Oxidation, basically oxidation, the most important is lipid. But you have to know oxidation also can be comes from the protein. Protein also can be oxidized. Okay. So the oxidative degradation of the pupa. This is the enriched by the fish, polyunsaturated fatty acid. It can cause the rancid flavor and also the odor and discolorations. Okay. So this is the phase, the autolytic spoilage. Okay, or the what is mean by autolysis is because as a self-digestion. Okay, the self-digestion is because of the enzyme breakdowns. Okay, enzymes active. Okay, and it can cause the spoilage happen. Okay, the digestive enzyme will break down the food consumed by the fish. Okay, and act as a biological catalyst. Okay, which is breaking down the muscle structure. Okay, this is the initial phase, and the progressive textural degradation leads to the softening of the flesh. Okay, so bila kita cakap softening of the flesh, of course dia akan reduce the texture quality. Okay. When reduce the texture quality, also we have few signs. Okay, for example, like rupture of the belly wall, okay, they either loss of the marketable quality, and of course the nutritional values also degrade. Okay, it's getting low. Okay, because of the spoilage happens. Okay. So usually the chilling, okay, like 0 degree slows the enzyme activity and the most effective measures lah to delay this kind of the spoilage. Okay, this is an example. Okay, how the enzyme okay, react. This you can see clearly. This is the enzyme. Okay, because of the peptides and free amino acids can be produced. Okay, so peptides and the free amino acids in the fish. Okay, when react with the enzyme, it can cause like this kind of the appearance, and also it can cause the belly bursting. Okay, belly bursting. Sometimes the belly bursting also because of the gas production in the fish. Okay, too much of the gas happens, the enzymes also actively helps, and then it can cause the burst in the intestine. Okay, this is the one that the questions given. Okay, the flavor changes. Okay, so this is the nucleotide degradations starting from the ATP, okay, right after that, okay, and then the AMP, ADP, AMP, and also they produce the IMP. So this is the most called the umami taste, and desirable sweet, milk, flavor taste, and very good, very high in fresh fish and so on, okay. But finally, afterwards, okay, when it comes to the inosine, okay, it comes to hypoxanthine, okay. So this is the critical, okay, because inosine it can produce the sour taste because it becomes acidic, okay. So the IMP degrades, okay, and becomes this is the IMP, and releasing of the phosphate, okay, and becomes the inosine, and then finally the inosine is acidic or sour, it becomes the sour, and then finally it becomes the hypoxanthine. Hypoxanthine is the bitter taste, okay, the bitter taste. Right. So this is the flavor precursor. Okay, this is can cause the off flavor of the product. This is the enhance the flavor. Okay, so but is it happen only at this stage? No, basically it will continue to degrade. Okay, up to the final product like hypoxanthine, and hypoxanthine also will more degrade into the xanthine and for the other compounds. Okay, so only encourage at this stage during this even the feed also they have the shelf life is when it is encouraged or recommend to be consumed. Okay, because we want the sweet taste of the flesh. And the third one is belly bursting. Belly bursting because of the enzymatic spoilage. This is our ability now. Okay, it can degrade quickly and spoils easily right after cough. Okay, why it bursts? Because of the presence of the carbon dioxide and also hydrogen gas. Okay, so where do you get the carbon dioxide and hydrogen gas? This is happen because the bacteria proliferate. Bacteria is producing the carbon dioxide, okay, and with the hydrogen gas, it can cause a radicals. Okay. And the final part is the color changes. The color changes is important, okay, because of the reaction of the enzymes and non-enzymic reactions, okay, and so on. Okay, yang may happen sometimes because the compositions in the flesh or the meat. Okay, the composition means that maybe because of the high heat, it can cause the very high also in the liquid oxidations, and this can affect so the color. Okay, so the color you can see here, the different colors. This is high, which is have the oxymyoglobins. Okay, the low myoglobins and also deoxy. Deoxy is the meat that has no oxygens. Okay, no oxygens. Okay, that's why the color is a bit darker here. Okay, so this is the flow. Okay, deoxymyoglobins, which is no oxygens before it cuts, and you make it a blooming. This is the process of blooming. When you cut the fish, when you cut the meat products, and you let it like maybe about 20 minutes, this is the blooming process. The blooming process is where the oxygen is mixed or functions to enhance the color, to improve the color of the products. And so they have the myoglobin, is whereby this is the problem effects because of the ions is oxidized and so on stuff. And the bacterial actions, how bacteria enter this and spoil the fish. As we know, the bacteria are not present in flesh. But they are found in skins and so on. And finally, they will invade. They will invade through the gills, kidney, blood vessels, and finally invade through the muscle tissue. Okay, and then after finally it can cause the spoilage. Okay, spoilage, and of course definitely because of the toxic metabolites. So these are examples of the bacteria, common bacteria like usually, as I can say, salmonella. Salmonella basically from bacteria is common to happen to the chicken. Okay, it can cause the diarrhea, food poisoning, Staphylococcus aureus, and also the Pseudomonas species usually have presence of the bacterial actions. Okay. So this is the bacterial presence. Okay, you can see it causes slime, skin, intestine. That fish is called the spoilage. Okay, this is we can found and detect the Pseudomonas presence. Okay, on top of the fish flesh. There are three types of bacteria. Basically, the first one is the spoilage type of bacteria, saprophytic bacteria. Saprophytic bacteria is the bacteria found in the tropical species organisms. Okay, the Pseudomonas and so on. Okay, and the second one is we have the pathogenic bacteria, pathogenic and indigenous and non-indigenous bacteria. This is the difference. Okay, this is just a spoilage. Okay, this is the pathogens. Okay, pathogenic. Okay, if severe, it can cause to death. Okay, either indigenous or non-indigenous. But indigenous is more distributed in the aquatic environment. Okay, the mine in fresh fish, but severe when multiplication growth, like Clostridial and so on, Vibrio and Aeromonas. Kalau when we talk about non-indigenous, okay, it comes from the polluted aquatic environments, okay, from the fishes, handling during the harvest and so on. Okay, macam Salmonella, Shigella, and E. coli, this is during the handling, and this is the bacteria that present in the environment, that present in the water. Okay, usually it's a minor problem. Okay. But this is, you have to be careful with this bacteria, Clostridium botulinum. Clostridium botulinum is dangerous, okay, because the spores, okay, and the Clostridium yang akan menyebabkan severe effects, and starting from the diarrhea, food poisoning, diarrhea, and so on, and finally, if there is no action, it can cause to death. Clostridium is, there are several cases happen, Clostridium botulinum. So the final part is the oxidations. Okay, how oxidation spoil the fish. Okay, you can see the contact with the fat or the pigment fish tissue. The fat oxidation comes produces the cell rancid of odors. Okay, the pigment oxidation, and it can cause the results in the unpleasant odor and discoloration. Okay, this is the flow of the oxidations. So oxidation basically is usually comes from the lipid, okay, and the proteins. But this is why I emphasize on the lipid, whereby the lipid meet with the oxygen. It can come to the lipid hydroperoxides. And lipid hydroperoxides, it is unstable. When it comes to the unstable, it will break down into the aldehyde and ketone. This is the late stage. Okay, this secondary degradations. Okay, this is the primary. Okay, primary degrade. This is secondary degrade. Okay, right. So this is the flow. I think I can share this slide later on to the England and might share to the students. Okay, this is the flow about the parasite value. The parasite value comes from the primary, this one, hydroparasites. Okay, but it is unstable. Okay, and then this blue parasite, because it's unstable, because usually the degradation happen rapidly. Okay, from the lipid it degrades to the hydroparasite degrades to the melandialdehyde and so on, MDA. Okay, so MDA is the second lipid oxidation of the pupa. Okay, so MDA usually kalau kita nak check ataupun we want to identify the lipid oxidation, we will check based on this MDA. Okay, and melandialdehyde. Okay, so melandialdehyde MDA, how we want to check the MDA is through the thiobarbituric acid reactive substances, because the chemical, the TBA chemicals will react with the MDA. Okay, because of the reactive oxygen species ROS. Okay, can help in we want to measure, okay, the ROS with the byproduct of the lipid oxidative image. Okay, so that's why we need the catalyst, some sort like the catalyst or the reagents, okay, to react with the MDA. Okay, so the MDA is the stable component compared to the parasite, hydroparasite. Okay, but for information, hydroparasite also can be measured, but the result is not accurate. Okay, I have to be honest that the result is not 100% accurate because it is unstable compounds. Okay, compared to this MDA. But MDA, you have to use the reagents, okay, to measure on the reactive oxygen species happen or present, okay, during that stage. Okay, why we focus on the lipid? Because the lipid oxidation, okay, it has a visible yellow brown discoloration, and this can cause the respiratory and odor. Okay, so this is, you can see the type of the fish: white fish, oily fish. Oily fish has a much more high values of protein lipid oxidation process. Okay, mentioned. Okay, this is the how it happens. Okay, they have the how the activations happen. Okay, it can be from the heat, the light. Okay, the light means that this is the what we call the preventions as well. Okay, and the metal ions, because of the trace amount of copper and iron, have this on the pro-oxidant catalyst. Pro-oxidant, you want to reduce or suppress the oxidation process. Okay, kenapa you nak suppress? Because you tak nak further oxidation. Because further oxidation, it can cause the off flavor, off odor. So that's why you need something to what we call it, suppress the oxidation process. Okay, to break down or to slow the oxidation process. Okay, and of course the moisture also can contribute to the oxidation. Okay, high moisture of course can promote the oxidation. Okay, by facilitating the oxidation contact. So this is the influence the aquatic produce species, as I mentioned, agent sites, environment, and also the seasons and foods. Okay, this is the protein composition of fish flesh, usually about the protein. This is common sense. 60-31%. This is the dry basis, and also this is the this is the wet basis. So this is the liquid. Okay, it can be as low as 0.2 to 25%, because it depends on the diversity of the liquid. Okay, the fat content. Okay, higher fat has the faster liquid oxidation and shorter the shelf life. Right. The off flavor of the fish, it can cause from the causative organisms, bacteria, okay, that present in the aquatic environment, and also it can caused by the compounds. This is what we call as jasmine. Jasmine, it smells like the earthy smell, something bau tanah, musty. And MIB, liquid residual is about the volatile compounds produced by the microorganism. this is the serious problem when we talk about the freshwater species. Sebenarnya freshwater species ikan nila, lele dan sebagainya, they have the contribution of the high jasmine and MIP. Okay. So this is why we are trying to reduce the this blending. Okay, ataupun reduce or kill this compounds, this jasmine and MIP, because they bau tanah, they bau freshwater. So that's why we are thinking about this alternative way. And also control during the culture, okay, minimize the feed waste, okay, regular water exchanges and so on. And finally is the processing lah. Processing is value added and comes to the control the temperature. That is the extended shelf life of the product. Okay, the final part, okay, sorry, take the time. Okay, basically the final one is the excess fish quality. Okay, we have three types here. We have the destructive method. We have how you use the samples and destroy the product. Okay, we have to analyze, lab testing, masuk dalam lab. Okay, homogenize the samples, then you process and so on. Okay, this is called as a destructive method. Non-destructive is the advanced technology that assess the quality without damage the product. Non-destructive usually use camera image. Okay, we use the spectroscopy, okay, to assess the quality. And the final part is the sensory evaluation based on the human. This is more on the qualitative. This is the quantitative, this is qualitative based. So the destructive, of course, is the multi-basket analysis. I think you are familiar. Deploying the correlate the test count, okay, you count the bacteria manually, okay, and the chemical analysis use the chemical quality indicators to analyze, and the physical is done by the texture analysis and so on. Okay, okay, this is the most important, the non-destructive method. Why? Because we don't want any damage of the products. We want to save the products. Okay. And also this because the non-destructive is non-invasive. It is safe. You don't need the use of the chemicals, you don't need the use of the any of the reagents, okay? Accurate, rapid and also the user friendly. For example, like electronic nose, electronic pump, okay, that is a mimic the human taste and smell. Hyperspectral and multispectral image is for the capture the spectral and spatial information. This can detect the freshness, detect the compositional changes, and also detect the any of the compounds present. Okay. The near infrared NIR, nucleotide magnetic resonance NMR spectroscopy also is a rapid chemical pre-analysis that can identify the marker compounds, compositional changes also in the finished product. And finally is the computer vision. This is common use in the industry. Okay, they have the conveyor belt here. Okay, the image analyze algorithm access the color, surface texture, and morphological changes for the quality. Okay, this is my what I did. Okay, previously on using the hyperspectral image is a non-destructive method. Okay, this is a sample. You capture the image, okay, from the samples, and the samples, okay, you integrate into the data, transform into the image visualization, and from this image visualization, it can form into the data. Okay, so how can you compare the compositional changes and so on? So from this data, they provide the quantitative results. Okay, from this quantitative data, they can compare dengan pada library. Kita ada database. Okay, database from where? From the lab testing. We open the lab testing to justify the range of the results. Okay. So kenapa kita focus on non-destructive? Because the first is, of course, this is much more easy. Okay, because usually the traditional the lab testing is the time consuming. Okay, that's why we introduce to this kind of the non-destructive method. Okay, so easy to analyze. Okay, it's non-invasive plus the product came from the lab testing and so on. So that's why we are introducing to the non-destructive method. And even right now, most of the industry the company, okay, the supply chain, they also use the non-destructive method. That's why they have the conveyor belt tu, yang ada conveyor, they can detect the spoilage and also the fracture. Sama juga kalau macam sekarang ada apps, okay, apps take the picture, okay, dia ada inform this is spoilage or this is the fresh samples, okay. And the final part is the sensory evaluation. This is from the human. This is the qualitative data. Then when we talk about the qualitative data, of course, it is subjective. So that's why it is subjective. You have to make sure that the panel should be either the trained panelist or the untrained panelist. Kalau trained panelist is okay. Kalau trained maksudnya sudah melalui beberapa fasa training, they can be like up to 10 panelists is more than enough. Okay. Tapi kalau untrained like us, okay, students or consumers, that can be at least 50 panelists, 50 panelists untrained panelists to check the evaluation of the product. Why? Because it is a subjective. Maybe your smell is my smell is different. Maybe interpretation. Alright? So these are examples of the form of sensory evaluations. Okay, they have also the QIM, quality index method. Okay, they macam mana skin, blood, skin, blood, and so on. They have the characters and score. The score can be varied. Yeah? It can be 0 to 3, it can be 7, it can be 9. But usually 5 or 7 score. Okay, because if you want more than that, usually because it can cause the variations of the value. Tapi kalau itu like the maximum is 3, is minimum, itu low score, it can affect some bias lah later on. So I think that's all from my part. Okay. So if you have a question, please feel free to ask.
Baik adik-adik, silahkan kalau ada yang mau bertanya. Ahmad mau bertanya.
Hello, Mr. Aswar. My name is Ahmad Sofian. I'm from Yogyakarta, Indonesia, and studying in Fisheries Gajah Mada University. I'm sorry, I don't know. In Malaysia, your country, most people use mixed language English with Melayu.
Okay.
Yeah, I want to ask in English, Mr. Aswar.
Yeah, possible. It's possible.
Okay.
Yeah, you can ask.
And what are the factors that cause contamination factor in fisheries product? Asia and Indonesia mostly use a traditional method in process with treatment for fisheries research. Factor for contamination.
So you're saying the traditional analysis, right?
Yeah, correct.
Yes, we can. Betul lah. This one. Okay, assessing the fish quality, right?
Right.
Okay. Okay. So basically, right now, the commonly assess the fish quality is destructive. Okay, even in Malaysia also, the university usually we focus on the destructive methods. Okay, why? Because sometimes the non-destructive method, we are still developing the optimal. We are trying to find the optimize the value. Okay, so how you optimize the value? You still make a database. So database comes from where? Database comes from the lab. Okay. In Malaysia also we are still doing the university also we are still doing the destructive method. Okay, practical. Okay, because we want the students to hands-on to see how to evaluate, how to track the microbiological, the PCA, okay, the strip cleaning, okay, and the chemical analysis, how to track the based on the check the readings of the spectroscopy or readings of using the refractometers and so on, and also the physical, okay, we check using the analyzer. So we want the students to apply give hands-on, okay, so that they can more understand and more clearly observe this kind of the testing compared to the non-destructive. Okay, so usually the non-destructive industry they currently apply for this one because the industry they don't want to do okay, they want to prepare the samples for the lab testing, okay, and also of course they want to because the non-destructive is non-invasive, okay, and the chemical usage and also non-destructive, so that you can use the product, okay. Even though we say okay, we just take a one batch, just one batch sample, but the thinking one gram we can still sell the product, okay. So there is the sample, we can still sell the product and we can make the money. So why do you want to waste the 1 gram product for the lab testing? Okay, so they want to fully utilize the product. Okay, so that's why they are focusing on the non-destructive methods. Am I answering the questions?
Thank you.
I think you also asked about the factor of contamination.
Yes.
You would like to know what is the factors that make the products contaminated, mainly for the traditional products.
Okay, alright. So the factors of contamination basically, the first one as I mentioned is the handling. Okay, the handling. Because the contamination can be two types of the contamination: the biological contamination, the physical contamination, and also the chemical contamination. Okay, okay. Let's see the easy. Okay, the physical contamination is where you handle the product without glove. I think they have the protocol when handling or making the products, okay, without the glove, for example, or without the hand wash properly. There's no biosecurity, okay, of the farm, okay. So that is a physical, okay, when we touch. And then second is the the biological contamination. The biological contamination is based on the cross contamination. Okay, when you, for example, you buy the seed, particularly seed of the fish, or when you supply okay, any of the materials, okay, and so on. That might be the cross contaminations that will happen. Okay, and the third one is the chemical contamination. The chemical contamination because of the enzyme start because of the stress. So because the fish is stressed, of course they produce a lot of the slime, and also the chemical contamination whereby the nucleotide degrades, okay, and also they produce the biogenic amines, the histamines can be triggered, and so on. So there is three factors of the contaminations, and the most contamination usually I can say about the bacterial contamination, the biological contamination, and the physical stuff, the rough handling, and because of the temperature, effects, and so on. Okay.
Alright, thank you, Mr. Aswar.
Okay. Any other question?
Ada lagi mungkin? Ada, silahkan.
Di sini aja.
Sebutkan namanya ya. Agak keras.
Okay, good morning, Mr. Ahmad from UMP. My name is Charlotte. I'm from Fishery Product Technology Study
Not really, but
Program.
I will try to listen. Okay, alright?
Okay. Okay, good morning, Mr. Ahmad. I'm Charlotte. Oh, sorry. I'm sorry, Mr. Okay. My name is Charlotte. I would like to ask, but in Indonesia, jadi kan ikan berdaging merah itu diketahui memiliki kandungan mioglobin sama zat besi yang lebih tinggi. Nah, sehingga dapat memicu pembentukan ROS atau senyawa-senyawa ROS yang memicu juga peroksidasi lipid. Nah, namun apakah pembentukan ROS dan peroksidasi lipid ini juga memungkinkan terjadi jika terjadi di ikan yang berdaging warna putih? Nah, jika iya, apa metode yang paling tepat untuk mengetahui deteksi kemunduran ikan pada ikan yang berdaging putih itu. Begitu. Thank you. Sorry, Mister.
So could you please start because I couldn't hear clearly.
Okay. So Carlos asked about the red meat and white meat with Dr. Asma. So because of the red meat, it has higher content of iron and other components that make it easily to be oxidized. Is it also happen the oxidation in the white meat? And how to measure the
Yeah.
quality of the white meat in
Method.
terms of oxidation?
Okay, alright, thank you. So yes, I can say that red meat has high potential in terms of the oxidation compared to the white meat. Okay, because the red meat has the iron, and also we have to know the red meat also contains of the myoglobin, high myoglobin. Okay, that can cause or trigger to the ROS can have a reactive oxygen species, and also it can contribute to the spoilage. Okay, that's the thing. But somehow, but still, okay, if the white meat, okay, it also has a potential for this oxidation process. It still can be happen basically, but the contributions of the potency of the red and white meat, okay, the red meat has a more potential, okay, of with more like high oxidation process compared to the white. Okay, you have to know the white meat also can be happen of the oxidation because the white meat they also have the liquid. Okay, so the liquid also can affect with the ROS can also produce the spoilage. Okay, cuma kalau what we want to say, what we want to justify basically the red meat has more tendency of the rapid oxidation compared to the white. Okay, but please you understand that both can happen the oxidation. Okay, it can both can happen the oxidation. Just the high the rapid oxidation or the slow oxidation process. Okay, and so both white meat also lakukan proses the oxidation. Cuma the time, okay, the period of the oxidation process might be lower compared to the red meat because of the presence of the iron, the presence of the myoglobin with the oxygen, and then also can be triggered with the ROS. So every of the animals, humans, everything ada melakukan proses oxidation. Am I answering the question? What is the factor, right? The second question.
She asked the measurement whether is it the same to measure the oxidation in red and white meat.
Yes, correct. It is the same actually to measure the red and also the white meat. Okay, but the result might be slightly different. Okay, because usually the value of the red meat is higher, a bit higher compared to the white meat. Similar with the result of the color might be different. Kan kalau you guna colorimeter tu, the color of the red and also the white meat is different. Okay, similar with the result of the oxidation, it might be slightly different. Okay, so if you want to compare, it's better you compare the red meat result with the other red meat, other species. Okay, so for the white meat, you can compare with other white meat species. So I think this is the more accurate. If you want to, I don't encourage you to share or to compare red meat and the white meat results because they might be slightly different lah in terms of the result. So kalau sama, the prosesnya sama, the agent pun sama, but the result basically is a bit different lah because the value is a bit different.
Oke, Caca menjawab.
Terjawab Ibu, terima kasih.
Sama ya, dia punya teknik prosedur sama ada.
Thank you, Mister.
Ada lagi mungkin pertanyaan sisi kanan? Sisi kanan saya ada yang mau bertanya? Ada belakang juga boleh. Di ditampilkan di kelas ada belly bursting tadi ya, belum sempat kalau pada saat di sesi saya belum tersampaikan. Kemudian tadi kita juga ada metode non destruktif, itu tadi kita juga ini ya, wawasan baru gitu ya, belum mungkin ada dia belum saya mendapatkan gitu ya dari selama di kelas. Ada mungkin yang mau ditanyakan? Kemudian tadi kalau ada menyimak ada juga geosmin, MIB tadi
MIT.
sempat terdengar nggak apa efeknya pada
Umami.
ikan?
Umami.
Jadi ada beberapa hal yang saya rasa melengkapi gitu ya apa yang disampaikan di kelas. Silakan kalau ada yang mau
Oke.
ditanyakan.
Aku SS kok. Ini.
Kalau bisa saya tahu ada ide.
Boleh, silakan.
Oke. Kalau di sini lah di Malaysia kita yang Yusmin dan MAP itu memang itu dua compounds yang sampai sekarang kita cari solusi. Oke, kita cari solusi karena kalau untuk fresh water memang ini kandungan yang tertinggi. Oke, jadi kandungan ini yang menyebabkan yang efek bau fresh water ya, bau kayak bau rumput, bau tanah itu, bau tanah itu. Jadi saya coba cara guna alternatif bagaimana mau menghilangkan bau. Mungkin dari segi mungkin kauternya mungkin beda ataupun mungkin dari segi handling dan prosesnya gimana ya. Jadi sampai sekarang pun kita coba ya mencari yang correct way ya, dan protokol yang mengurangkan bau di handling ini.
Ada yang mau ditanyakan? Silakan. Mau tanya? Enggak. Jadi oxidized product, and then which one we should assess for the oxidation, maybe by using or by using PPA or maybe there is a different stage of oxidation that we can assess. Can you get the question?
Sorry, the
This
thing
one.
I
This
cannot
one.
really.
Okay, so as you mentioned previously in your slide, there are several steps in oxidation process, like the early stage, and then we have the primary and the secondary product. So when we would like to assess the quality of the lipids in fish products, which one or in what condition that we have to measure whether we determine which kind of parameters of oxidation, because they have the early products and also the late or the secondary products.
Okay, alright. Thank you for the question. This is very interesting. Now, why? Because now there are two methods. Okay, the first one is total peroxide value (TPV). Kalau you can check total peroxide value, and another method is the unity box. Okay, this is both methods measure the liquid oxidation. Okay, but it depends on the people or the research. Okay, but basically we focus on the secondary product, which is the T-bus. Why? Because the T-bus, is more the T-bus is more stable compared to the peroxide. Okay, the total peroxide value. Okay, and then because yes, you can get the value for the TPP, total peroxide value, but sometimes the accuracy is less compared to the T-bus. Okay, the T-bus is more stable and it is easily to be measured compared to the TPP, total peroxide value. Okay, but in a general way, it depends also the what kind of the target. Okay, what kind of the products and what kind of the target. If you are measuring the meat, the fish, DBOS value order based on the melon NDA. Okay, the NDA. Okay, NDA is the secondary products. Okay, but if you are measuring on maybe on other food products, like non other food product, maybe you can use on the TPV, total parasite use. Okay, so actually it depends on what kind of the product, the species you measure, and also what I encourage with the fish, it is better to measure based on the NTA. Okay, NTA which is using the DBOS value. It's more stable and the data also more accurate. Okay, because with the parasite you have to make it rapidly and you have to make it faster. Okay, because the degradation is going to be rapid degradation as compared to the DBOS, okay, which is the secondary, which is getting slower of the degradation.
Is there any other question? Last question, Dr. Aswad. You have mentioned about the non-destructive methods. I think it's a very interesting methods, but mainly they will be used by a big company, right?
Yes,
Because if
big
you
company.
and how accurate those non-destructive method compared to the traditional method? Because when we use the traditional methods, we can directly know about the value, those that oxidation process and other chemicals, which is the parameters of the deterioration. But when we use the non-destructive, it looks like it's a non-direct measurement. Yeah, probably you could explain how accurate or yeah, how far the results from the traditional.
Okay, alright. So, so when we talk about how accurate of the non-destructive method, basically, it depends. Okay, in the non-destructive method, it depends on the AI that help in processing the data. Okay, so the AI they have a lot. We learn about they have a machine learning. Okay, they have a deep learning and so on stuff. So the AI, even right now, also they are using the two types of the deep learning. Okay, to get the most accurate data of precision value. Okay, so if you want to measure, you want to know the accuracy, we have to measure the precision value. Okay, so the precision value, if given deep learning on different software, it should be near to the one. But usually, for example, what I tested before, I used the genetic algorithm, okay, and also the deep learning. The accuracy of my non-destructive, my camera image, like this one, okay, I use with the software. Okay, right. So this one, I use the software, okay, I from the image and they transform into the data. So the data, I use the software of the genetic algorithm and also the machine learning, okay, to sort, okay, the important information, okay, and I get the result. The accuracy is between 0.7 to 0.95, okay. So accuracy is if nearly to one is more accurate compared to the lesser than that. And also, for information, also we have tried this kind of the camera image towards the composition, the compounds. It's not only the texture, it's not only the general, but also deep inside the composition. Okay, kayak okay, kita check on the liquid oxidation, in NDA-nya, how what is the value of the NDA. Okay, but if you want to check on the NDA and so on, we have to get the testing first, the lab testing, the data, what's called the database first. Okay, so that we can compare. Okay. So for the industry, we have a database. Okay, they run several times of the lab testing. So they have the the database, and they compare with this kind of the non-destructive. So when they have more than 0.5 or nearly to 1, up to the nearly 1, so they have to optimize this kind of the non-destructive method. So they use this one as a, for the next regular of the testing quality testing afterwards. Okay. So when we talk about the accuracy, it depends on the software, it depends on the machine learning that we are doing, and also even right now also they are improving type of the machine learning, like in deep learning, and also type of the processing system of processing data. We have the pre-process, we have the processing data. Okay, because this processing data we discuss unrelevant information or unrelevant data. Unrelevant data comes from where? Sometimes here it can come from the light, the light, the outside. Okay, sometimes get from the vibrations, okay, the moisture. So we are going to discuss this and get only the data that is for our target compound lah, or target component. Okay.
Jadi kita bisa tahu kualitasnya secara real time begitu ya, secara langsung,
Betul.
tidak perlu menunggu lama ya.
Even right now kalau untuk aquaculture pun kita ada IoT ya kan. So IoT also the real time monitoring. That's why the IoT also like this company lah. Meaning that they have the sensor. So from the sensor they akan give the data, and the data the real time can give the signals to the farmers. Okay, so it can be also as an early detection system lah. The farmers also actually is very convenient. Okay, but somehow to develop this, we have also need to try and error lah to get the optimize the software and also the kind of the machine learning that we use.
Terima kasih sekali Dr. Azwar atas kuliah yang disampaikan. Mungkin adik-adik boleh testimoni setelah mengikuti kuliah Dr. Azwar, apa yang dirasakan, boleh disampaikan ke Dr. Azwar. Siapa mau? Ahmad atau yang lain? Siapa?
Really interesting.
Hari ini saya kurang enak badannya.
Oh, boleh. Semoga lekas sembuh Dr. Azwar. Dita boleh menyampaikan sesuatu?
Really insightful. Thank you so much.
Insightful. Thank you so much. Yeah. Thank you so much,
I hope
Dr.
that
Azwar.
I hope that you can understand my lecture, and I try to make it simple presentations. And if you have a question, then you can just ask me anytime. Okay? All right. Wish you all the best.
Thank you very much. Thank you very much. So we really hope that you can visit us sometimes in the future, and we will have another collaboration. I hope that we can do further collaboration. Thank you very much. Wish you all the best. Sukses selalu, Dr. Azwar. Baik, adik-adik kita boleh sampaikan terima kasih begitu ya ke Dr. Azwar.
Terima kasih.
Love you.
Foto bersama Dr. Azwar.
Ini first students profile in the end. Di sini
Adik-adik, ini adik-adik yang di sini.
ada absensinya.
Biar keluar dua ini kayaknya masih
Saya
oh gini ya. Oh iya, di mana kabar?
Kita foto ya. Sini. Linknya udah. Iya sebentar ya. Bes.
Yang lagi online boleh sambil dinyalakan nggak? Ini yang di sini online.
Sarah tapi kan
Udah. Mau ikut.
nggak kelihatan.
Sebentar.
Yang pada
Sebentar. Oke. Ya udah sih. Nah. Turunin ya. SMP lah. Bentar. Nah, kelihatan nggak?
Siap ya. 1, 2, 3. Sebentar, saya capture dulu.
Iya, mukanya burung-burung.
Kasih jempol ya, Dede, kasih jempol.
Enggak kali.
Terus ya.
Jempol. Kasih MM formal lain.
Oke, ya.
Ya udah.
Wajah hilang. Oke, satu lagi. 3, 2, 1.
Close the door.
Oke.
Gasnya habis.
Semuanya Wide, terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Amin, amin, amin.
Salam.
Jangan lupa ya, ada jangan lupa isi ya resum dan isi atan dan usus ya Bu ya yang di persepsinya. Sudah ada di oke.
Mana, Bu?
Siapa nih? Saya ya.
Pesta babi.
Ini linknya. Udah dapat.
Tag masih siapa sih?
Duh, habis baterai lagi. Ini ya. Ada yang bawa powerbank nggak?
Ya ini yang kelas ini dulu aja. Tapi tetap ngisi Google Form ya. Kadang Google Form aja ada.
Oh, mau Indun dulu.
Ini semuanya ya, Bu?
Ini yang kelas ini dulu, yang kelas 209.
Hampir aja sih.
Ini kelas 209.
Duh, habis baterai lagi. Nggak ada.
Jangan
Susah.
Cunlay udah belum?
Hah?
Jangan Cunlay.
Yang apa?
Kemarin yang mikro
Apa?
udah bikin summary.
Udah yang 209.
Aku minta materinya dong, ada nggak?
Ada sih materinya.
Itu yang kelas 209.
Kita kelas bersusah. Cara aku kirim.
Minggu depan ada satu sesi lagi internasional dari Vietnam. Daerah Dede terbiasa mendengar, hari ini kan dari Malaysia gampang ya. Kemarin dengar dari Cina ya.
Susah.
Ni hao ma.
Iya itu. Susah apa prima? Susah. Beneran.
China.
Internasional ya Dede. Baru papan internasional.
Waduh,
Sudah yang 209.
globalisasi ya.
Oke, sekarang kita ganti yang 309.
Aduh, 1%.
Mau ikut ya, Lap?
Hah?
Absen selfie sama aku.
Emang ada?
Iya, aku kan ada absennya harus selfie. Oh, kalau itu ada besar.
Oh iya?
Iya, serius.
Segitu ya.
Nah, bisa. Failed.
File. Aku bisa kok. Mau
Itu kayaknya ibu harus scan dulu deh.
Iya, biar jadi.
yes.
Belum di scan belum. Belum di scan sama ibu. Itu lah itu loh.
Di grup THP.
Gue udah nggak tahu.
Paling bawah ada nggak? Iya, iya sih harusnya studi ini apa? Studi banding ke Malaya.
Kebacet. Bener lah. Aku kelas siapa ya? Udah enggak apa-apa diisi dua-duanya. Diisi dua-duanya, Mas. Bacot.
Nanti untuk resume kita buka di Simaster gitu ya, jadi silakan resumenya diupload di Simaster masing-masing kelas. Baik, kalau sudah, nanti yang belum silakan sambil akses gitu ya, kita akhiri. Terima kasih sekali. Insyaallah kita ketemu lagi minggu depan kelas terakhir sebelum kita ujian gitu ya. Dicek lagi adik-adik untuk presensi untuk ujian boleh ikut UAS kalau minimal 75% gitu ya, segera dicek segitu adik-adik. Kalau ada yang error presensi dan lain-lain silahkan disampaikan ke akademik gitu ya. Baik, terima kasih lagi Ustaz. Insyaallah ketemu lagi pekan depan. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Thank you so much.
Belum diisi. Udah ada saran boleh nggak sih? Hah? Ada saran? Nggak wajib ya? Oh iya.
Assalamualaikum tidur.
Habis ini tuh tidur habis resepsi.
Tidur pulang aja jam. Jangan anjir tasnya cuma
Kasih ngomong.